Good day everyone. So the topic assigned to us is the relationship between turgor pressure and tissue rigidity.
First, we discuss about the introduction in the relationship between turgor pressure and tissue rigidity. Turgor pressure is a key component of plant physiology. It is the internal pressure exerted by central vacuole against the cell wall. This pressure is maintained through the osmotic flow of water into the cell. The turgor pressure is central to several critical plant functions, including maintaining cell shape, tissue rigidity, driving cell expansion, and assisting in water uptake and nutrient transport. So in other words, turgor pressure is internal pressure against the cell wall. This pressure maintains cell shape, supports tissue rigidity, drives cell expansion, and aids water uptake and nutrient transport. In essence, it is crucial for plant structure, growth, and overall physiological function. So next, turgor pressure. Turgor pressure is a pressure exerted by the fluid, usually water, contained within the central vacuole of a plant cell against the cell wall. It results from the osmotic flow of water into the cell due to the difference in solute concentration between cell's vacuole and the surrounding environment. So, turgor pressure is like the internal push in a plant cell, created by water moving into a large storage area called the central vacuole. This pressure helps the plant stay upright, keeps its shape, and take in nutrient water efficiently. Next is tissue rigidity. Tissue rigidity refers to the firmness and stiffness of plant tissue, which is essential for maintaining the structural integrity of plants. Rigidity is particularly important for plant cells to withstand mechanical stress, maintaining their shape and support the plant's overall structure. So tissue rigidity in plants is like their body strength. It's how firm and sturdy plants are to handle things like wind or weight. This firmness helps plants keep their shape and supports the plant's structure like natural backbone. So next topic is about methods of turgor pressure measurement. To understand the relationship between turgor pressure and tissue rigidity, it is crucial to utilize accurate measurement methods. So first method is about pressure probe. To measure pressure probe, first insert the probe to a chosen plant cell. Adjust the probe to maintain constant position inside the cell. As the water moves in or out the cell, the pressure changes and record these changes. Higher pressure indicates the cell is full and firm. Lower pressure suggests that the cell is losing water and becomes less firm. So next method is about ball tonometry. To measure using ball tonometry, gently press the tonometer against the plant cell. Measure how much force applied and divide it to the contact area of the ball and the plant cell to get the pressure. If the plant cell is full and firm, the tonometer will push back less. If the cell is losing water, the tonometer will push back more. The tonometer gives you a measure of how much resistance the plant cell has. More resistance means higher turgor pressure. So the next method is indentation technique. Picture the plant cell as a soft pillow filled with water, representing turgor pressure. Gently press on the plant cell with a tool like poking a pillow with your finger. If the cell is full and firm, it resists indentation, meaning it has higher turgor pressure. If the cell is losing water, it indents more easily, meaning it has a lower turgor pressure.
So next is the role of turgor pressure in tissue rigidity. Turgor pressure is directly linked to the rigidity and mechanical strength of plant tissues. When plant cells are fully turgid, they exert pressure against the cell wall, resulting in a firm and rigid structure. Turgor pressure is essential for maintaining the structural integrity of non-woody plant parts, such as leaves, stems, and flowers. The pressure keeps cells turgid, preventing wilting and collapse. So, makompare natin ang Turgor pressure sa isang balon na pag mapuno na ng hangin yung isang balon is nagiging firm or turgid na yung surface niya. So, next is the cellular and structural mechanisms. The relationship between Turgor pressure and tissue rigidity is governed by several cellular and structural mechanisms. So first is the cellulose microfibrils. Cellulose microfibrils in the plant cell wall provide structural support. Turgor pressure pushes the cell membrane against the cell wall, and the rigidity of the microfibrils resists this pressure, contributing to tissue rigidity. Second is the osmotic flow of water. Water uptake into the central vacuole through osmosis increases Turgor pressure, resulting in cell expansion and increased tissue rigidity. The loss of Turgor pressure due to water loss can lead to wilting and reduced tissue rigidity. So, for the conclusion, the relationship between Turgor pressure and tissue rigidity in plant cells is a fundamental aspect of plant physiology. Turgor pressure maintains cell shape and tissue rigidity by pushing the cell membrane against the cell wall. This pressure, combined with the rigidity of cellulose microfibrils, ensures that plant tissues remain turgid and structurally sound. A comprehensive understanding of this relationship is essential for the study of plant growth, development, and responses to environmental factors. That's all. Thank you for listening.